Thank you very much for this opportunity. Yes, indeed, today I'm going to talk about extracellular vesicles, in this case, EVs derived from bacteria, in this specific case from Lactobacillus, and their role of these EVs in protecting from HIV-1 transmission. So here it is very important to talk about the vaginal microbiota. Indeed, very quickly here, the mucosa layer of the female lower genital tract, it is normally colonized by several microorganisms, and they are in dynamic equilibrium with the host. Also, there were several studies done in order to characterize the vaginal microbiota of healthy premenopausal women. And most of these studies demonstrated that the vaginal microbiota is dominated by Lactobacillus species. Lactobacillus are considered also health-promoting bacteria because they prevent against the overgrowth or colonization of several uropathogens. In particular, it has been already shown that Lactobacillus can prevent against HIV transmission. We also already proved that Lactobacillus isolated from healthy premenopausal women can prevent from HIV-1 transmission in human tissues ex vivo. So based in this previous work, in this specific project, we wanted to understand whether the EVs or extracellular vesicles derived from this Lactobacillus are actually the ones responsible of this anti-HIV effect. Very quickly also here about the EVs. EVs are secreted by any kind of cells, and they are important for cell-to-cell communications because they can carry several cargos such as proteins, nucleic acids, metabolites, and etc. So we basically, what we did is we cultured overnight Lactobacillus strains, and then after by sequential centrifugations and ulterior ultracentrifugation, we were able to isolate EVs. Then these EVs were characterized by nanoparticle tracking analysis or nanosite according to their size. For example, these 4 Lactobacillus strains secrete EVs of similar sizes of around 137 nanometers, and they secrete particles ranging from 10 to the 10 to 10 to the 11 particles per ml. With these EVs, we tested their anti-HIV effect. Indeed, what we did is we treated HIV with bacterial EVs, and after we infected human CD4 T cell lines, in this case MT4 and Jurkat, we also infected human tissues ex vivo, in particular cervicovaginal and tonsillar tissues ex vivo. Then the HIV replications were measured in the cell or tissue culture medium by measuring the P24. Here you can see the results of this anti-HIV effect. In this case, are MT4 cells infected with HIV-LAI and then treated with EVs from 4 Lactobacillus strains. So you can see here that the EVs derived from Lactobacillus crispatus BC3 and Lactobacillus gasseri BC12 decreased HIV replication significantly. We took these EVs that decreased HIV replication strongly, and then we tested their concentration effect. So in this case, we took these EVs derived from Lactobacillus gasseri BC12. So we used the MT4 cells infected with HIV-LAI and then treated with different amount of EVs. So you can see here that the effect of this anti-HIV effect is in a concentration-dependent manner, starting actually at 5 per 10 to the 4 particles per ml. We confirmed these results in using different cell line, in this case, the Jurkat cells infected with different strain of HIV val. And in similar way, you can see here that the effect of the EVs in decreased HIV replication is increasing actually the amount of EVs. So these results demonstrate that indeed EVs derived from Lactobacillus can prevent from HIV transmission in human cell line. At this point, we wanted to test in a system that mimic in vivo conditions. So we used human tissues ex vivo. These are, for example, we used human cervicovaginal tissues. This is the surgical specimen obtained from the routine hysterectomies. But basically what we did is we separated the mucosa layer from the stromal tissue. And then these stripes of mucus, we cut in small blocks of 2 to 3 millimeters. And then we transfer 9 blocks per well into the surface of the gel phone. Then we culture these tissues ex vivo for up to 15 days, replacing the medium every 3 days. In a similar way, we use tonsillar tissues ex vivo. So we separated the necrotic or bloody tissue, and then we cut and culture like cervicovaginal tissues ex vivo. Here are the results of this anti-HIV effect of bacterial EVs in tonsillar tissues ex vivo. So you can see here that the treatment with EVs derived from Lactobacillus crispatus BC3 and Lactobacillus gasseri BC12 decreased HIV replication significantly, similar to what we observed in human cell lines. We took also the EVs derived from Lactobacillus BC12, and then we tested also their effect, increasing the amount, a different amount of EVs. And here also you can see that the tonsillar tissues infected with HIV-LAI, you can see that the effect is also in a concentration-dependent manner. We tested also the effect of these EVs in cervicovaginal tissues ex vivo. And in similar way, you can see that indeed the effect of the EVs is also in a concentration-dependent manner. So with these results, we can say that EVs derived from lactobacillus can prevent from HIV transmission in human cell lines and also in human tissues ex vivo. So at this point, what are the bases of this anti-HIV effect? In this case, we have to answer 2 questions. First, whether the EVs are doing something specifically on the host cells, or these EVs are doing something on the virus itself. To answer those questions, we ask a trivial question whether the effect that we observed was because of the cell cytotoxicity. So in this case, we used different cell lines, and in this case, MT4 cells and Jurkat cells, and then we treated with the highest amount of bacterial EVs, all these 4 lactobacillus strains, and then we we measure the cell viability. So you can see here that the cell viability in both cell lines were not altered in any case. This using the propedion iodide-based assay. We observed similar results in the MTT assay, and we don't see any alteration of the cell viability. We confirm also these results by flow cytometry. We look at the CD3 and CD4 population in EV-treated cells compared to the control experiments. So we don't see any changes. And yeah, these results just demonstrate that indeed EVs were not cytotoxic. On the other hand, testing the effect of these EVs on the host cells, to understand whether these EVs are doing something on the target cell, basically what we did is we used the TCMBL cells. We treated for 24 hours with bacterial EVs. And after 24 hours, we wash off these cells to eliminate any remaining bacterial EVs, and then we infected with HIV. So you can see here that the pre-treatment with bacterial EVs in any of the cases didn't prevent from the HIV infection. Similar results we obtained also using the MT4 cells treated in the similar way. So we can see from these results, we can say that indeed seems that these bacterial EVs are not doing anything specific on the host cells in order that they are producing or secreting any antiviral compounds. So yeah, but still we need to understand what is these EVs doing on the virus. Why we have this? We observe this decrease of this HIV infection. So we wonder whether the HIV viral entry or attachment into the target cells was altered. So in this case, to study the HIV entry, we use the TCMBL cells. We infected with HIV-LAI, and then we treated with different amount of EVs from Lactobacillus gasseri BC12. So you can see here that the HIV entry in TCMBL cells was decreased significantly, increasing the amount of bacterial EVs. So this is telling us that indeed bacterial EVs are doing in somehow that the viral entry is decreased significantly. Regarding to the attachment of the virus into the target cells, to study this, we used the MT4 cells. We infected with the HIV-LAI, and also in the same time, we treated with bacterial EVs. This was done for 2 hours at 4 degrees in order that we block the viral entry. So doing this, we washed, of course, several times these MT4 cells in order to eliminate any free viral particles or free bacterial EVs. And then we measured the P24. So we can see that the treatment with EVs derived from Lactobacillus crispatus BC3 and Lactobacillus gasseri BC12 decreased significantly the attachment of the virus into the target cells. So these results demonstrate that indeed the HIV entry and attachment into the target cells is altered. But at this point, we wanted to focus then, these bacterial EVs are doing something specifically on the virus itself. So to study this, we did capture experiments. We basically, what we did is we used magnetic nanoparticles associated to PG9. PG9 recognized the trimeric form of the GP120 on the virus. So to do this, we treated the virus with bacterial EVs for 1 hour, and then after we capture with magnetic nanoparticles. So doing this, what we observed is that the amount of virus captured with Lactobacillus crispatus BC3 derived EVs and also EVs from Lactobacillus gasseri BC12, the amount of virus captured was significantly reduced. So these results demonstrate that EVs are doing something specifically on the virus itself. So they are actually affecting, altering the viral envelope, specifically the GP120. We also did a proteomic analysis by mass spectroscopy of these bacterial EVs. We found several proteins. We started doing the recombinant proteins of these EV-associated proteins. And then in this case, I'm showing preliminary results. In this case, we use the recombinant enolase and elongation factor 2. And we observed that elongation factor 2 is decreasing the HIV replication, as you can see here. So this can be one of the proteins responsible of this anti-HIV effect, but we are still working on all these other proteins. So with this, the conclusions, our conclusion is that the EVs released by lactobacillus protect from human cervicovaginal and tonsillar tissues ex vivo, as well as human cell lines from HIV infection. And this protection was not because of the cytotoxicity, but was rather associated with the decrease of HIV entry or attachment into the target cells, mainly by affecting the viral envelope. And these findings demonstrate that the protective effect of lactobacillus against HIV transmission, it is in part mediated by EVs released by these bacteria. And these bacterial EVs can carry numerous proteins that can be associated with this anti-HIV effect. And finally, I would like to acknowledge all the people involved in this project. At the NIH, Dr. Margolis, Dr. Bampuy, and all our collaborators, our Italian collaborators from the University of Bologna, and also all the core facilities at the NIH. Thank you.